So this chapter 6 is about the molecules that macrophages make and deal with the immune system. And these are some of the probably the most important molecules that the macrophages make, because they really set going a whole cadre of molecules that are called pro-inflammatory cytokines. And the story begins way back in the 60s, as most of the stories in this business have done, in 1968. So you remember in 1965 is when they first discovered these supernatants from activated lymphocytes had the so-called conditioned medium, had growth factors in them. Ultimately, they became called T-cell growth factor and then interleukin 2. Well, that was 65. In 68, there were two different investigators, one on the West Coast, one on the East Coast, that simultaneously really described a similar kind of thing, and that was supernatant molecules, soluble molecules that were in these conditioned media that would kill other cells. And the first was Gail Granger at UC Riverside, I think, in California, and then Nancy Ruddle at Yale. And what they both did was activate lymphocytes or mononuclear cells, whatever, with antigens. And then they took the supernatants, they cultured them for a while, they took the supernatants for them and they put them onto target cells, and they used fibroblastoid cell line that was called an L cell. And they could radiolabel the L cells, the target cells, and they could show that these supernatants would kill those cells because they would release their radiolabel over time. And you can, as you recall, also when I discussed cytotoxicity, Bruner and Serratini had described it also in 1968, the chromium release assay. I think these investigators used a different radiolabel and so forth, but it doesn't make any difference. The supernatants had something in them that would kill. And they thought, because of the way they made their supernatants and their activated supernatants, they thought they were coming from lymphocytes, and so they called this factor, this active ingredient, they called it lymphotoxin. And then, only a couple of 3 years later, 1971, there was a fellow in London by the name of Peter Alexander, and he was interested in macrophages. And so he took macrophage cultures and activated them with what are becoming the sort of the normal activators of macrophages, which were, on the one hand, LPS, lipopolysaccharide, the so-called endotoxin from bacteria. And he also used a molecule that I'd worked with around the same time, 1970, 71, called poly-IC. And that was a synthetic polymer of ribonucleic acid, RNA. And it was originally synthesized to mimic RNA viruses that had a genome that was made out of RNA as opposed to DNA. And he showed that they would make supernatants, and that was when then placed onto radiolabeled target cells, that this stuff would kill as well. It wasn't clear whether this lymphotoxin that had been discovered by Granger and Ruddle was the same or different or whatever, and so there it stood. And it wasn't until 1975 when an investigator by the name of Lloyd Old, and his portrait, is over my shoulder right here. That's a portrait of Lloyd Old, and he was an interesting fellow. In 1958, he graduated from the University of California, San Francisco, and medical school. And he graduated evidently first in his class, and his parents gave him a Corvette that he then drove across the country and to take up his postdoctoral fellowship with an investigator there. And so he just skipped altogether clinical training, and he went in directly from medical school into science. And he became established there. He was interested in killing tumors. That's what his whole thing was. And so everybody had known by then that if you that if you take mice and infect them with BCG, the Bacillus calmette-guerin, which is the TB vaccine, and it's a live bacterial vaccine, that it would sort of hop up their whole immune system. And also, what Lloyd Old did was he first infected them with BCG, and he'd wait until that sort of stabilized, and then he would inject them with LPS, lipopolysaccharide, 2 weeks later. And what he found was that the second injection with lipopolysaccharide would, within 24 hours, would cause the mice to go into shock. Their blood pressures would drop, and essentially they'd die, or they'd almost die. If they recovered, then they would be okay again. And what he wondered was that why was the blood pressure going down like that? Why were they going into shock? And so within that 24-hour period after the LPS injection, he bled the mice. He took the fluid portion of the blood, the plasma or the serum, and he injected that into mice that had large tumors growing, usually subcutaneous tumors. They put them on the leg or the back where you could see it easily. And he found that within 24 hours of injection of the plasma from these mice that had been treated with BCG and LPS, that the tumors on these mice would undergo what they called hemorrhagic necrosis, so that the tumors would just sort of, scab up and drop off, essentially. And that was pretty dramatic. And he decided that ultimately they did some biochemistry and some other kinds of assays, and they could show that it wasn't due to the BCG effect per se or the LPS effect per se, but the two together would markedly potentiate this whole thing, and that there was a new factor that the mice seemed to produce in the plasma, that then if you use that to inject in tumor-bearing mice, it would make the tumors go away. And that he named tumor necrosis factor, and that was in 1975, and it's a classic paper in our business. Subsequently it was shown that you could do the same thing with poly-IC that Peter Alexander had used. And you could also show that the serum from these mice that had this TNF activity in them, that it would kill cells in vitro in these assays with chromium release or with other radiolabeled release to show that the target cells were actually killed. And so immediately then Lloyd Old figured that was the active component, and it directly killed tumors. And that's one of the reasons they named it tumor necrosis factor. And they didn't have any idea that this had anything to do with the immune response or inflammation or anything else. And they didn't really care. They just wanted to show that they could kill tumors this way. Well, so that was 1975. And it wasn't until 5 or 6 years later, another fellow got involved in this whole process, and that was Anthony Cerami from the Rockefeller University in New York, right across the street from Lloyd Old, who was at Sloan Kettering. And then I spent a lot of my career at Cornell, which is right across the other street, and that's the Cornell Medical School. So I know Tony, and I met Tony early on and so forth. And so Tony, he was a biochemist, and during his early training, he had gone to Africa on a Rockefeller Foundation fellowship, and he got interested in these chronic infectious diseases that were causing wasting, because he said all these people that were infected, they looked like they were just wasting away. They were becoming, the medical term is cachectic. And same thing that happens with tumor patients nowadays. You just sort of waste away and you get really skinny and so forth. And it wasn't clear why this was happening. And Tony went back to the lab, back to the Rockefeller, and he decided to work on this. And he didn't want to have to work with the actual bugs that were causing this wasting disease syndrome. And so he decided that he investigated whether or not LPS would do it, because LPS was a chemical, essentially. It was a big molecule, but it was, you could buy it at the store from Sigma or wherever one of these biological supply houses. So he focused on LPS, and he injected them into mice, and he found that if you didn't kill them with the hemorrhagic shock that Lloyd Old showed, he would end up with mice that would become cachectic, just like the patients in Africa. And so when he investigated further, being the biochemist that he was, he looked at the serum, and the serum was filled with fat. It was hyperlipemic, is what we say. And so you hear the poor mice were wasting away, and they had more fat in their blood than they could deal with. And so he ultimately traced that down to show that there was a molecule on the surface of fat cells called lipoprotein lipase. And what lipoprotein lipase does, and he showed at the time, was that it would take the fat molecules that were floating around in the plasma and allow them to go inside cells. And what happened when they were injected with LPS is the lipoprotein lipase stopped working. So the fat just built up in the blood. And so the cells didn't have any fat to really work on. And that's why they became cachectic. And so he did the same thing then that Lloyd Old did. And that is he said, well, is it LPS or is it something else? Is it something that LPS stimulates the body to make? And that was the right answer, the second possibility. And so he did similar kinds of experiments that Lloyd Old had done, and showed that this was due to this factor that was activity that was in the blood, and he gave it another name. And his name was cachectin. And so, because the mice became cachectic. And that whole process, that whole story, from the initial in vitro cytotoxic activities released by both lymphocytes as well as macrophages, tested by the radiolabeled release assays, and then the in vivo assays of Old, and then subsequently Serotini, or Cerami, were where it was all at up until about 1980. And then, but this time in 1980, it's interesting in terms of the evolution of American science, because by this time, biotech companies had been started, and they arose from the mid-70s, primarily from gene cloning. And one of the first, if not the first, was a company called Genentech. And it was started in a warehouse in South San Francisco, sort of on the wharf, on the docks. And that company subsequently became very wealthy. And by 1980-81, they had really geared up, and they'd hired all kinds of really top-notch scientists that were academicians, and they'd offered them more money and so forth and so on. And so they built up a whole departments of protein chemistry, molecular biology, immunology, and so forth and so on. And there was a young investigator, Michael Palladino, who had been a postdoc with what's his name, Lloyd Old, from New York, and he was hired by Genentech, and he went about convincing everybody within the company that they should work on TNF because it was really important, or any of these molecules. And so they did. And they set up a department of protein chemistry, and one of the major players in the protein chemistry department was a Indian guy by the name of Bharat Agarwal. And they set up which was at the time in sort of an industrial approach to purifying, making and purifying lymphotoxin, TNF, anything they could get their hands on. And with regard to lymphotoxin, they found a lymphoma cell line, a tumor cell line, a lymphoid tumor cell line that could make lymphotoxin. They grew up 300 liters. You know, back in the day, we thought we were good with 100. I mean, not even 100, 100 milliliters, not liters. So they have 300 liters, and they ultimately condensed that and purified that down to what he could show. Pretty much so it was homogeneous, meaning that it was only one molecule there that had the toxic activity. Very soon thereafter, they got a partial amino acid sequence on that, on their purified protein, and then the gene jockeys got in. Their department was brought in, and they cloned the gene for that. And that was lymphotoxin, and they published that in Nature, and that was 1984. Then in 1985, they published another paper. This time they had purified what they ultimately called tumor necrosis factor, TNF. They started out initially with normal human cells that were activated with BCG and LPS, just like Lloyd Old had done. But ultimately they found a cell line that was called HL60, and they were able to grow up, 100 or so liters of that, purified it, and then and it had TNF activity, both in vivo, injecting into mice, making their tumors go away, and also in vitro, it would show they could show that it would kill these cells. So that was 1985, and that caused quite a stir in the community of budding young scientists such as myself at the time. But then Tony Cerami wasn't quiescent during these years, from his initial cachectin discovery in 1981. And a young postdoc came into the lab, whose name was Bruce Beutler, and oops, he's over here. That's Bruce Beutler right there, and these are both portraits that I've done. These are pastel portraits. So Bruce was basically a wunderkind. And he went to college when he was still a teenager. He graduated early. He went on to medical school at the age of 20 something. And his father was a very famous scientist at Scripps Research Institute in La Jolla. And he told Bruce that the best way to become a scientist was to go to medical school rather than graduate school. And I've made a video on that. And so Bruce did that, and he went to the University of Chicago Medical School. And then the second thing he told him, and I agree with this, is that you had to solidify that medical school experience. You had to go into the hospital and really see a lot of sick people and take care of them and be a resident. So he went to the University of Texas Southwestern to do that. Then he came out and he was a postdoc with Tony Cerami at the Rockefeller. And they set off together to purify cachectin. At the same time that the guys in Genentech were purifying TNF and lymphotoxin. And so ultimately, Bruce was successful. They purified enough of the protein that they thought was cachectin to get some amino acid sequence from it. And when they compared the amino acid sequence with the predicted amino acid sequence of TNF and lymphotoxin, they were very similar. And so that set the stage for a quest that was solved soon thereafter, that cachectin essentially was TNF. Well, that was interesting, because the next thing that that Bruce did is that they had purified enough cachectin to radiolabel it and to look for cachectin, or now I'll switch to totally TNF receptors. And lo and behold, what they found is that there were receptors on all kinds of normal cells, muscle cells, fat cells, all kinds of things, macrophages, and so forth. And so that meant that Lloyd Old's tumor necrosis factor wasn't just a tumor killer. It had something to do with normal physiology. If there were cells in the body, all over the body, that would have receptors for this stuff. And it put shed a whole new light on things, particularly for Cerami and Beutler. One of the first things they did, Cerami and Beutler, was to use their purified molecules and inject them into rabbits, so that rabbits would make antibodies to the TNF. And they were successful doing that. And they then took their rabbit antibody and they asked the question, if we mix together this antibody with LPS and then inject mice, will it prevent the shock? And lo and behold, that worked. And so that meant that it wasn't the LPS, it was the TNF that the LPS caused the host to make that ultimately caused the shock. And that's very, very important, because for the longest time, LPS, lipopolysaccharide, bacterial lipopolysaccharide, had been called endotoxin. There was an endotoxin associated. And people assumed that the toxin was LPS itself. But for the first time in science and also in medicine, now we knew that that the real endotoxin was tumor necrosis factor. It wasn't the LPS. And of course, in this time of COVID-19 and so forth, TNF is one of the major players, and what's causing all of the severe symptoms of these people that ultimately end up in the intensive care unit. They also did one more thing. They made lots of recombinant from the gene cloning experiments. They made TNF that way, and collaborated with a surgical group over at Cornell. And a young postdoc by the name of Kevin Tracy, injected rats with the TNF and showed that the TNF directly would cause the shock. So there was no LPS involved, and so forth. Now, the very next year, in 1986, Charles DiMorello, who is the fellow that I talked about earlier in the interleukin-1 story, well, that's also a macrophage product. And Charles knew that it caused fever. It was an endogenous pyrogen. And he wondered whether or not how that related to the TNF and the lymphotoxin and the cachectin and all this stuff. And so he collaborated with both the Cerami group and the Lloyd Old's group, and showed essentially that the TNF lymphotoxin when injected into animals, would cause the animals, would score positive in the rabbit fever assay that he had devised, number one. But number two, what it did when you injected the TNF, it caused the production of IL-1 and IL-6. And of course, IL-1 and IL-6 are two of the other pro-inflammatory cytokines that participate in the septic shock syndrome, and now in the acute respiratory distress syndrome that the COVID-19 patients are getting. So all of those experiments that were done now in the 80s, and so now how long ago was that? Was that 20 years ago or whatever. So that still has not trickled down necessarily to internal medicine. Early on, after the Cerami and Beutler had made an antibody to TNF, immediately people, and then they showed that it caused the shock, the septic shock. People wondered whether they could use it in the clinic for all these patients that were, and they're still coming in, by the way, all the patients that were coming into the intensive care unit with very low blood pressures and severe bacterial, usually bacterial infections. If they could just give them anti-TNF, maybe they could save these people, because the death rate, I think, the mortality rate is like 40%. And so they did a clinical trial and they tried it and it didn't work. So I think that put a kibosh on a lot of the clinicians in terms of buying into this cytokine business, you know, and it sort of dampened everybody's spirits. And there was a company that was developing this anti-TNF for the use in septic shock and so forth, and they ultimately were getting ready to shelve it. Until towards in the late 80s, an investigator by the name of Mark Feldman, who was interested in rheumatoid arthritis, asked them if he could try it to treat arthritis patients. And he first did it in a mouse model, and then they took it into the clinic, and the good news is it worked. And now the biggest selling drug in the pharmacy is Humira, which is a human antibody against TNF. So the biggest selling drug in the pharmacy today is Humira, which is a human monoclonal antibody that reacts with TNF. And I mean, it's a multi-billion-dollar drug, number one on the 10 best selling drugs in the United States. But the other thing is, the number 5 selling drug is Enbrel. And Enbrel is not an antibody, it's a soluble TNF receptor. And actually Bruce Beutler, who's over my other shoulder here, what's this one over here, Bruce Beutler, he developed that after he left Tony Cerami's lab at the Rockefeller and started his own lab. He developed that drug, which is now called Enbrel, and it's number 5 on the list. And then number 9 on the list is Remicade, which is not a human antibody but a mouse antibody. And that's the antibody. That antibody was generated by Jan Wilczek at NYU, and it's against TNF. All 3 of these drugs are against TNF, and they all are used to treat the same diseases. First, rheumatoid arthritis, then psoriasis, ulcerative colitis, Crohn's disease, and other inflammatory bowel disease, and so forth. Remicade was the antibody that was used by Mark Feldman when he first said, you know, let me try this for a chronic inflammatory disease rather than try to use it for an acute situation. And by the way, I should probably say a little bit about that. I think, so nobody really went back and tried to use anti-TNF to treat septic shock in an acute, emergency situation where the person's really going down the drain quickly. It was a very difficult clinical situation to try to test a new therapy in. Persons in the intensive care unit, they're really, really sick. They've got all kinds of problems and so forth. And so, but I think the major reason it didn't seem to pan out and work very well was the 2 reasons. One, the cat was out of the bag already. The TNF had been released and had been around for quite some time, and it had already done its damage by causing the capillaries to leak all over the body so that the blood volume retracted and the blood pressure fell. Yeah, the other reason is that TNF is not the only pro-inflammatory cytokine. There's interleukin-1, there is lymphotoxin, there's interleukin-6, and interleukin-6, I covered that a little bit when I was talking about the generation of antibody-forming cells, because one of its major functions is to help the differentiation of B cells into plasma cells so that they can make huge amounts of antibody. It's a differentiation event in the B cell compartment. So so if you just use anti-TNF in the shock kind of a situation, you're not blocking IL-1 and you're not blocking IL-6 or lymphotoxin and so forth. And so that needs to be revisited, particularly in the times of the COVID pandemic, because it's the same pathophysiology. It just in COVID, it happens to be very acute in the lung first, because the lung fills up with inflammatory fluids, and then you die from that. And so, but that was for chronic inflammation. Acute inflammation sort of died with the antibody to treat septic shock. So the other group of molecules that the macrophages make that are really, really important are the interferons. And I have some personal stories about interferon that I'll also tell you here. So the interferon was basically one of the first cytokine discovered, which was in 1957. It goes back before the 60s. By two fellows from Europe, Isaacs and Lindemann in Switzerland. And they found that if they infected cell cultures with a virus and then waited a while, washed off the virus or whatever, got rid of the virus, but then took the supernatant from these cells and put it on the new cells, and then challenged the new cells with virus, that it would interfere with the virus. So that's why it was named interferon. So that was 1957. And between 1957 and the 1980s, so over a 20-year period, interferon had its ups and downs in popularity, but ultimately became a legitimate molecule. And that story is important because now it's being used in the clinic. So in the beginning, the virologists were interested in interferon because it blocks viruses. And their favorite cell to use in tissue culture was a fibroblast, and it would form, the fibroblast would attach to the bottom of the flask, and they would grow and cover the bottom of the flask, and then you could put virus in and you could see that where the virus was that was killing the cells, essentially. And that was basically the interferon assay. The virologists, being virologists, worked up a very nice doubling dilution. They would titrate the virus, and then they'd also titrate their interferon samples, and they would put those two together, and they could quantify then the amount of interferon they had in the whole thing. Then in 1965, same year that the mitogenic factors were discovered in lymphocyte culture supernatants, there's a fellow by the name of Fred Wheelock from the University of Pennsylvania that found interferon activity, antiviral activity in those same kinds of supernatants. So fibroblasts could make it, lymphocytes, monocytes, they didn't know which one. And then a fellow by the name of Kari Kendall from Finland, who was the head of a blood bank in Helsinki, and had his hands on large quantities of donated blood. He could purify the red cells from the white blood cells, and he could take the white blood cells, mix them together with viruses, and then look for interferon activity in that supernatant. And he started then trying to purify interferon from huge quantities of blood bank kinds of cells. And what they found early on, that you could use virus to activate the blood cells. Then you can also use this phytohemagglutinin that would cause the lymphocytes to proliferate. And then you could also use specific antigen, provided the lymphocytes came from a person who had been vaccinated or sensitized to that particular antigen. So that took us from 1957 to 1970. And in 1970, an investigator in France, in Paris, at the CNRS, which is sort of like the NSF in our country. It's really scientists who focus down on fundamental basic things, a lot of physics and mathematics and so forth, but they do also do biology. It's a fellow by the name of Agressor. And he showed that interferon preparations that had interferon antiviral activity, if incubated under tumor cells, leukemia cells, they would block the proliferation of the leukemia cells. Well, that just, blew the lid off all the oncology people, as they said, we've got the answer to cancer, and we can use that to treat all kinds of cancers and so forth. So that was 1970, and then in 1972, Agressor showed that the same antiviral interferon preparations would block the proliferation of lymphocytes, the kinds of lymphocytes that I had studied activated with PHA or specific antigen and so forth. So it was not only was an antiviral factor, it was an anti-proliferative factor. And that's sort of when I got into the business, because I left the NIH in 1972 and went to Dartmouth Medical School to train with a fellow by the name of Ross McIntyre, who was interested in interferon, and he was a hematologist. And so early on, so I learned how to do one of these doubling diluted interferon assays, or how they did it. I wasn't trusted to do that in the laboratory yet, as I was new. And then we took the interferon preparations that we were working with and so forth, and we could show that I showed that they would block the proliferation of the lymphocytes in the same way that Agressor had already done. And then Ross had a clinical trial ongoing at that stage, where nobody had any interferon. They couldn't treat patients with interferon because it was in minuscule quantities. And so people started looking for interferon inducers. Well, we could use LPS to induce interferon. We could also use poly-IC. So people wouldn't want to inject LPS into people. But Ross got an investigational new drug application approved to use poly-IC to treat patients. And so along came Kendall, and so I was sort of assigned to administer the poly-IC to these patients and take their blood and watch them over them so that hopefully they wouldn't go into shock and so forth. And that was one of my first clinical investigative experiences doing that. And what we showed is that in fact, when you gave them an injection, an IV push injection of the poly-IC, and then followed their blood over time, that there would be a peak of interferon activity that would come up and then would go down. And when the interferon activity was at its peak in vivo, we could take blood from those patients and look at the lymphocytes, and we could show that their proliferative capacity was not as good as it was before we started, or then afterwards, after the interferon had gone away. And that was interesting. And about the time that we completed those studies, I left Dartmouth and I went to work with Georges Maté. He's over here. Georges Maté, who is right across the street from Agressor. And so one of the first things I did was look up Agressor when I got to Paris. because I figured, better meet this guy. And it was in 1973, when he showed that not only would these interferons inhibit tumor cells and lymphocytes from proliferating, they would stimulate the appearance on the cell surface of treated cells of the gene products from the major histocompatibility locus, the MHC encoded molecules, which are the ones, since the early 70s, that 10 years later were found to be the antigen, where the antigen would bind to them, and then that would stimulate the T cells, the T cell antigen receptor. So he showed that class 1 MHC was increased, and then a few years later, another investigator, Joe Oppenheim at the NIH, showed that class 2 was also augmented by interferon. And so this interferon seemed to be a really interesting molecule. It would inhibit viruses, it would inhibit cells from proliferating, it would stimulate gene expression on cells. And as time went on, other people had shown that it would activate the macrophages. So it got to be called a macrophage activating factor, or MAF. And so there was a lot of interest in it. And in fact, there was a lot of interest by the cancer people to get this stuff into the clinic and so forth. But it took a long time. It took most of that decade of the 70s. And early on, there was a lot of enthusiasm, and then it waned and so forth, because it didn't do what people would hoped it would be, the answer to cancer, and be a major hit. And just parenthetically, ultimately, as time went on into the 80s and the 90s, it was found to be active in chronic myelocytic leukemia, or CML, and also a disease called polycythemia vera, which is a precursor leukemia of the red blood cell precursors. So, but that took another 10, 15, 20 years. And in the meantime, interferon was sort of forgotten by the clinical people. So 1980 was a huge year in the interferon story, because suddenly the transition was made from an activity in culture supernatants to actual molecules. And the first breakthrough was done by a couple of fellows by the name of Lee Hood and Tim Hunkapiller at Caltech. And the reason that it was a breakthrough was that Hunkapiller and Hood developed a new machine that increased the sensitivity of sequencing proteins from one end to the other, multiple fold. So now what you could do is you could take the minuscule quantities of really pure interferon from all of your biochemical separative procedures, and you could start finding out what the sequence of amino acids is on one end of the chain. And that allowed them to identify an interferon with a particular sequence. And then almost simultaneously, Catherine Zune at the NIH, she collaborated with Hunkapiller and Hood because they had the technology, and they sequenced another molecule that she'd been purifying, and it turns out it was similar, but it had enough of a difference that they knew it was a different molecule. So now we have two interferons. And then Charles Weissman was a fellow over in Zurich, and he got together with Kerry Cantel that I talked about, who had the blood bank, huge quantities of leukocytes, white blood cells from the blood bank. And they purified and cloned the gene, the cDNA, the complementary DNA, for another interferon. And then another fellow by the name of Tada Taniguchi, who had postdoced with Charles Weissman when they were in the process of doing this initial cloning. He learned all the techniques. He went back to Japan, started his own lab, and then came up with another interferon. And then Charles Weissman came up with 8 more genes. And so suddenly there were a whole bunch of these interferons. And I still don't think it's been really figured out why the evolutionary, the Darwinian natural selection has thought it necessary to have so many different interferon molecules. They're all about the same size, and they all have very similar sequences, and they all react with 1 or 2 different receptors that was subsequently found out on different kinds of cells and so forth. But what interferons are is that they are very important in the so-called innate immune response, particularly to viral infections, because as soon as a virus comes into the body and takes up residence in some cells and starts to multiply, and then gets released, and it starts to spread locally and then systemically throughout the body, that causes the cells to release interferon. Well, COVID-19, for example, has genes that code for molecules that block the production of interferon. And so what happens is with the SARS-CoV-2 is that it blocks the innate immune response. And so that's why you get this 14-day period of no symptoms, because the symptoms are due to the cytokines, really. And that's been blocked by this virus until finally the T cells say, there's somebody here that shouldn't be here. And that takes 2 weeks for the T cells to wake up and start to react and so forth and so on. And so anyway, the innate immune response, and in particular interferon, is very important for our host defenses. Now, and one of the things I wanted to tell you was go into a little personal story about interferon in the clinic, because I don't know, maybe 10 years ago now, more. Yeah, 10 years ago, 10 years ago. I came down with some red spots on my back. And of course, I didn't look at my back very often. My wife told me, she said, you got some red spots on your back. And so I went to I had always been worried, because when I was a kid, I was a lifeguard for 6 years through high school and college. And I worried that I'd someday come down with melanoma, malignant melanoma, which is not a good disease to get. I went to my dermatologist. I got biopsied, and the biopsy came back positive for what they call cutaneous T-cell lymphoma. So I had been working for 40 years on T cells, and I get a T-cell lymphoma. I guess that was Mother Nature's way of telling me that I'm, got to watch myself. And so what I did then was I went to another doctor. I had another biopsy. I had that repeated. It came back with the same kind of reading. I sent the cells off to other people. The slides off to other people, and it came back with the same thing. And I knew, because I've been in the business and I started out in hematology and oncology, and of course I knew what cutaneous T-cell lymphoma was. It also has a word of mycosis fungoides. Mycosis means fungus, because in the early days, back in the 19th century, they thought it was a fungal infection because it causes funny looking rashes on your body and so forth. So I knew that it wasn't a major, it wasn't a major disaster. A rapidly growing kind of thing that I had to deal with. And so I sat down and I started to read. And so over the course of about 3 or 4 months, I must have read about 600 papers. And on the basis of the papers that I read all these things over and over and over again, and one paper would lead to 5 more papers, and then I read those, and it would just kept on going. And finally, I got the gestalt that 2 things. One is, because it presented in the skin, it was by default, the patients went to dermatologists. It was a dermatologic disease. Well, dermatologists don't do clinical trials by and large. They just treat with different ointments and salves and so forth. And so that's what they did with T-cell lymphoma, cutaneous T-cell lymphoma. It's abbreviated CTCL. So, and that, because it was a very slow growing process, their salves would work, the spots would go away or diminish markedly, and they'd have the patients come back in a month or 2 or 6 and so forth. And if it recurred, they would give them more salves or switch the ointments to another variety, and then they started mixing that in with chemotherapeutic agents in the salves and so forth. Ultimately, though, the patients would the salves wouldn't work anymore. So at that point, the dermatologists would throw up their hands and refer them to the oncologist. So the oncologist would then take these people. Then they did the clinical trials. So they had, but they had clinical trials. But the thing was that all the patients were so-called stage 4. They had it all over them. They had disseminated disease. Well, they tried all kinds of chemotherapy, and a lot of them would work a little bit, but then they would grow back and so forth and so on. And it took a while to accumulate a lot of clinical trials. But overall, what I noticed is that when they used interferon alpha, they said they reviewed papers or reports of multiple trials with interferon, and they got an 80% response rate. The response rate could have been a 50% reduction, that's called a partial response, greater than 50%, or a full reduction, and that was a complete response. And together that was 80% of all patients treated. And I said to myself, that's pretty significant. And so then, but by all my reading, I had identified a half a dozen people around the world that were dermatologists or oncologists who were experts in CTCL. So I called them up, introduced myself, and the good news was that they took my calls. They'd heard my name, through the literature or whatever. And they all told me the same thing. They said, Kendall, go low and go slow. And at that stage, I was all ready to come out with the biggest guns I could get to kill all of these bad guys as much as I could. But I listened to them, and I identified a physician in in Denmark. And I called him up, and I chatted with him and told him what I was all about and what I was worried about or what I was thinking about doing and so forth. And that I was thinking about using interferon, and he was one of the doctors that had used interferon to good success. And he said to me, I invited him to come to New York. And he said, you don't need me to come all the way to New York. You've got Dick Silver there. And of course, I had met Dick Silver when I was a hem-onc fellow or a research fellow at Dartmouth back in the early 70s. He was in Cornell as a hem-onc clinician, and I used to go to all of the clinical trial meetings, and I met him there and so forth. And I knew Dick Silver very well, and he was a superb doctor. He was old school, very old school doctor. So I went to him and I told him I had this CTCL. He made me show him my spots. And then after I did that, he said, now you're my patient. But I didn't understand that really. He says, no, and if I become your doctor, you're going to have to listen to me and do what I tell you. So I said, okay, that's a deal. And so we then embarked on a course of interferon, and he'd used it for 20 years to treat polycythemia. And one of the things I left out about my earlier study, when I was in Paris, I had done studies on erythropoietin and cells that were responsive to erythropoietin, and I had shown that interferon would inhibit the proliferation of the erythropoietin responsive cells, and erythropoietin causes red cells to proliferate. Well, and Dick Silver had used that in the clinic to treat patients with erythroleukemia. So he had a 20-year experience, clinical experience with using interferon. And the thing about interferons, particularly when used in cancer therapy, it was used chronically over a long time period. And the problem with it was people would come up with ultimately accumulate more and more side effects. One of the major side effects was that it would activate their brain and their mind, and they would get, a lot of them would get agitated depression, sort of road rage, to the point where they had to stop working because he was trying to kill his coworkers, or the people were, and so forth. And so I knew I didn't want to do that. And so, and Dick Silver didn't want to do that either. So he reduced the dose by, I can't remember, it was a half or a quarter. I think it was a quarter of what was used usually. And I was good with that. And so we started off on interferon, and after 2 or 3 months, the spots started to go away. And ultimately, over 4, 5, 6 months, they went away altogether. And so then we stopped the interferon, and we waited to see what would happen. And they never came back. And that was 10 years ago. So I know that interferon will work in certain diseases. And as I said, it worked for chronic myelocytic leukemia, works for polycythemia. Now we know that it works for CTCL. And so that's an interesting sort of anecdote, that in clinical medicine, it takes a long time to sort through all the variables and the vagaries and so forth, and also to get the basic science to be appreciated and used and become standard of care. So I'll stop there in terms of my stories.